We have Dr. Dustin Loy, or it says J. Dustin Loy, with us here this afternoon, and he has some new research and a vaccine out. So with that, Dustin, I was going to turn it over to you. There you go. Perfect.
All right, great. Well, happy to be with you today. I'm Dustin Loy. I'm from the University of Nebraska-Lincoln. I'm in the School of Vet Medicine and Biomedical Sciences. I'm a veterinarian and a microbiologist, and my primary appointment here is I work in the diagnostic lab. So a lot of the things that the vet panel talked about today with diagnostics, we're sort of intimately familiar with, and down in the trenches with y'all's clinicians and veterinarians, and trying to help them get diagnostic information. So I got asked to talk today about Pinkeye, so I'm gonna kind of just jump into it. This is kind of familiar, I think, to a lot of you. What Dr. Gillespie talked about with bovine respiratory disease. It's what we call a disease complex, and so by that I mean it's complicated. So it's not our typical, we have a introduction of a pathogen and it causes a disease and we treat or vaccinate around it. There's a lot of contributing factors, and so I'll go through that and kind of talk about what that looks like. Pinkeye is a disease complex and how we might think about managing it in our herds. And what was said previously about BRD, most of those things hold true for Pinkeye, unfortunately, making it a complicated issue. As I know, Pinkeye is one of the most frustrating diseases we have. My own cattle have dealt with it. Since I was a kid, we've always had Pinkeye. We haven't figured out how to get rid of it in Nebraska, and I imagine in Nevada you deal with the same problems. And because of that, we got to wear a lot of different hats. So I'm going to talk about some of those hats, everything from entomology to immunology to microbiology to cattle management to animal science. I think all these things factor into when we think about Pinkeye. First, I'm going to cover what is Pinkeye, right? So the etiology means what is the causative agent or factor that causes the disease. And we'll talk about that and what we know about that. We'll talk a little bit about flies. We have flies in Nebraska, as I imagine you do too. And so we'll talk about flies and how they're involved in the disease itself. We'll talk about vaccines, and Stacy mentioned we did some vaccine studies in our research herd. Like the university out there, we have an ag experiment station, and we're actively researching solutions for producers both in Nebraska and across the U.S. And so we're trying to figure out ways to help that. And then I'm a microbiologist, so my interest is in the bacteria. And so I'm going to talk about, we've done a lot of work sequencing these bacteria, and the veterinary panel mentioned Moraxella bovis. So thank you, vet panel, for bringing up my favorite bacteria. And so we'll talk about that and what we've done working on Moraxella. I mentioned this paper. This is the first paper describing Pinkeye in America. It's from 1889. It was from the first professor that started our department here at Nebraska. And you can see from those pictures before they had photographs, any of you that have cattle that have had Pinkeye, these will look very familiar, right? We have a central ulcer with new neovascularization or new blood vessels coming in. And they reported these in Nebraska. Unfortunately, we kind of are in a similar spot to where we were then, and we don't have great solutions, but we are trying. And I'll talk about what we know and what we can do about Pinkeye. And what we've learned over the last 125 years. Okay, so this will look familiar to those that heard Dr. Gillespie's talk. I don't have a fancy figure designer, so mine's a bunch of circles, but it's the concept still the same, right? So remember he had those gears in his. We kind of have a similar relationship between the different components of disease in Pinkeye. The first one's the host. So the host of Pinkeye, we're obviously at the Cattlemen's meeting today, so we're talking about cattle. The host contributes a part to this disease, along with the environment, and then the pathogen or the organism, the bacteria, all kind of contribute. And all three of these things need to be at a particular level of susceptibility. for the disease to occur. Okay, so we can't just target a bacteria, we can't just target environment or genetics of the host. All these things need to be kind of considered when we think about disease. So some things when we think about the host, we've talked a lot about genetics. I heard lots of information today about EPDs and marbling and carcass quality and heart failure. And pink eye is no different. We know that there's some genetic susceptibility to this. Traditionally, it's been white-faced cattle, so I love Herefords as much as the next person, but there is some information that shows that Herefords, because of that white pigment, they get more things like UV light exposure to the eyes and may be more susceptible to this disease than other breeds. Additionally, there's genetic sort of resistance to the disease. So Indicus cattle or Brahma breeds tend to have more resistance. And we don't see a lot of pink eye and Brahmas in general. And so they have some resistance to it. Other more to individual factors are things like stress and hydration. Remember, the eye, we're dealing with a mucosal surface where that liquid barrier is very important. So if we have stressed or dehydrated cattle, those barriers can be compromised and they may be at higher risk for things like pink eye. Vaccination status, we heard all about vaccines earlier today. Vaccines are important, not only for pink eye, but when we think about respiratory disease or other viral diseases that can cause immune suppression. We heard things like BVD, persistent infection. Those are all important things to consider, as those viruses can all contribute to making the animal more susceptible. And then calf source, right? Do we have to ship them all the way across the country? Are they going to be stressed? Are they coming from somewhere that may be at higher risk? The environment. This is the one that I think is hardest to manage in some cases. We haven't gotten rid of dust in Nebraska, and I bet Nevada you haven't either, right? So dust is a big problem. That abrasions on the corneal surface make these very susceptible to pink eye. I go out and work our research cattle multiple times a year. And those days it's real windy and dry. I have pink eye in the morning from all the dust that's gotten in there. And so you can imagine those little calves have a problem too. UV light. Irritates the surface of the cornea and makes them more susceptible to the disease. In eastern Nebraska, we have a lot of plant awns and seed heads that get into our forages that can irritate the eye and get stuck in there. And then in southeastern Nebraska, we have things like fescue that are sharp. The blades are sharp and can cause damage. I'll talk about flies, and flies are an important component of the disease. And then we have concurrent and underlying diseases, new introduction of new strains of bacteria, biosecurity. Do we have a vaccine coverage? Is it, you mentioned not only the vaccine composition, but the timing. Are we getting all our vaccines for respiratory disease? What might that risk be? And then pathogen burden. So we've done a lot of research looking at when the animals shed these bacteria, kind of what the timing is of those, what those look like in our microbiome research, in our research herd here in Nebraska. So I get asked by producers and veterinarians quite frequently why we have these really terrible outbreaks of pink eye some years. Others we don't have any. We did a 5-year trial in our research herd. Some years we had none. Other years we had 70% of the calves get it. So we get huge variation year to year. And I've kind of come up with this model that tries to account for some of that. And it really just integrates all those things that we talked about earlier. So we have calves that are susceptible, right? That are susceptible to pink eye. They don't have a lot of immunity. We have cows that carry the bacteria. Dr. Gillespie mentioned like respiratory disease bacteria that really they never get rid of it. And it's the same with pink eye bacteria. We're always going to have cows that carry those in the nose. We're going to get sort of another layer of other agents. So summer pneumonia, we may have something like a coronavirus or a respiratory infection in those calves. When those calves get about 2 to 3 months of age, those antibodies that Dr. Gillespie mentioned that are so important start to go away, right? So as those decay, the calf needs to start making its own, and there's usually a lag period where that occurs. So they're going to lose the antibody to not only pink eye bacteria, but everything else. Okay, at that same time, at least in Nebraska, we're getting summer temperatures coming up. We're getting a lot more UV. It tends to start drying out, so we get dust. And then at the same time, we get these pastures that go into that early production, and we get long seed heads in our pastures with a lot of irritating forages. As we move further into the summer, we get flies that start to emerge. Those flies are going to irritate them and cause fly avoidance behaviors. So all the cattle will circle, right, and they start swatting each other in the face to keep all the flies off. And that's a great way to kind of spread these bacteria from cow to cow as they're in close proximity. We get new strains. And the bacteria themselves need to turn on all their genes that start making toxins, right? So as those pass from calf to calf, they get hotter, right? So that means they're more likely to cause pink eye if they get in a susceptible host, because they really have all those toxins, those bad genes that they're making that damage cells ramped up and turned on. And then we get the outbreak, right? That's when the snowball kind of crashes down the cliff and we're at, treating half our calves. So this is kind of what's going on in the pastures when this occurs. What is pink eye? I thought it's important we cover this because pink eye is kind of used as any term of any eye disease in a cow, right? And I hear all sorts, they got pink eye or this one's got pink eye. But scientifically, when we talk about pink eye, it's a specific disease caused by a specific organism. Typically, this is Moraxella bovis. So this was work done by my mentor, Dr. Doug Rogers, when he was in graduate school. And what they did is they took calves in bubbles and they inoculated them with the Moraxella bovis bacteria, and then took samples and looked at them under the electron microscope. You can see that when those calves that are susceptible are inoculated, here's those bacteria. They have a protein called a pili that allows them to stick very intimately to the eye surface. Okay. Within a couple hours, those bacteria are secreting those toxins I talked about that are damaging and disrupting the surface of the eye. You can see the pitting and corroding that's starting to occur. Within another couple hours, they've almost completely buried through the corneal surface, and they are in that in the corneal layer, and they have migrated from the surface here almost all the way through. And then by 12 hours, they are all the way through. They are in the globe of the eye, replicating and causing a lot of damage and disruption in that eye. And then by one day, 24 hours, you can see how these turn white into sort of our classic pink eye lesion. That's the white blood cells that have migrated into the eye to try and get rid of these bacteria, right? So we go from just in one day, how quickly these can occur. We talked about, how to identify and treat animals rapidly. That's important. You can see how fast this can occur, and it can be really, really surprising how fast that can happen. On the flip side, the healing part of the eye is very, very slow, slower than we can possibly even think. So these are slides from Dr. John Angelos. He's at UC Davis, and they deal with a lot of pink eye in the foothills. The Sierra Nevadas out there by Davis and their herd. You can see that this is documenting the healing of an eye. This calf was treated here on day 0. It was treated effectively, and basically all the bacteria were killed with an antibiotic. And then all this is just the healing phases that they go through. So I hear all the time, that we have a non-responsive pink eye or pink eye that the treatment is just not working. And usually when I get pictures or look at those calves, they're somewhere in this healing process. The bacteria are gone. It just takes that long for the eye to heal. So remember, the eye doesn't have any blood vessels. The calf has to grow those new blood vessels. Those blood vessels have to come in to grow. You have to get new division of the cells that cover the eye. And then those cells and scar tissue have to resolve, until, several, more than 2 months later, we still have a scar in the eye from this healing process. So what causes pink eye? We talked a lot about Moraxella bovis. This is the one that we know we can prove in the lab that it causes it. But in our cases that we get, again, this is a disease complex, we get all sorts of findings from our diagnostic cases. So I'll show you some slides that we find. Everything from another Moraxella bovis's cousin, Moraxella bovaculi, Moraxella ovis. We get a lot of mycoplasmas, which are a really simple bacteria that doesn't have a cell wall. And those are often associated with respiratory disease with Mycoplasma bovis, but we also find those in the eye. You can get things like chlamydia, and then herpes viruses are a common one that can cause pink eye-like disease. Here's what we see in our lab in Nebraska. So we have a panel, a PCR-based panel, where we look for the presence of all these different bacteria in the virus. You can see that almost 90% have both Moraxella bovis, Moraxella bovaculi, and Mycoplasma bovaculi found in those cases. And you can see that the levels of these, as they go up, there's fewer, but there's quite a few of those bacteria in all these cases, with some Mycoplasma is almost 100%. We find very few associated with herpes viruses, so I think those vaccines work very well and people use them aggressively, and so we see very little ocular involvement with those. So how's pink eye maintained and spread? We get, again, I mentioned carrier animals, so animals can shed pink eye, at least 2 months after they've been infected. Usually it's more. They can carry it for months and months in the nasopharynx. So they get it in the nose and it kind of hangs out there for a long, long time. I get a lot of calls about winter pink eye. We don't have any flies. It's winter. Why are we getting pink eye? And those calves are usually in a feedlot setting where they're, nose to nose in a bunk and able to exchange those bacteria and cause that. So, we get these face-to-face contact, but in the pastures, it's primarily face flies that are going to transmit it. We did a lot of our microbiome type work, trying to figure out when these bacteria colonize and if there's ways we might be able to eliminate them from the animals, but really we couldn't. So we vaccinated them. We didn't see any impact on the presence between animals that got vaccine and that didn't. So that didn't help the carrier state. We saw that our young calves, so those ones that are 2 to 3 months old, had high levels of Moraxella. And then by the time, 3 months to 3 weeks later, and then at weaning, you can see those levels really decline, by almost, by 2 to 3 fold decrease in those. But at the same time, we see an increase in Mycoplasma. So we have shown that these bacteria are quite widespread. Their Moraxella is highest in the young calves when that maternal antibody is decreasing, and then that kind of decreases over time, where we see Mycoplasma increase. And then our vaccine protocol did not affect the carriage of those, so they're really hard to eliminate from the cattle. Transmission, the face fly again is one of our big challenges. These are in cow manure and pasture. They have about a 15 to 25 day life cycle. They're a challenge to control. They have these really abrasive bottle brush mouth parts that irritate the eye. And we've shown in some data done in the 60s and 70s showed that if you double your face fly burden, you double the risk, right? So there's an intimate relationship between the number of face flies and the amount of risk for pink eye. So one solution is fly tags. These are one of the most effective way to reduce fly burdens. So these all have an organophosphate, a pyrethroid, or a combination. If we are using fly tags, remember we want to make sure we remove them so that the flies don't become resistant. You want to use 2 tags for best results. So we got 2 eyes, so we need 2 tags. And we want to get those applied when we traditionally have had the most flies. So timing is critical. We want to get them on not when that fly burn is going to be maximal, and not too far before, because the chemicals decrease in efficacy over time. If you want more information on flies, David Boxer, our UNL extension educator, he's got a lot of information on his website. These are some other late season face fly controls. So if you didn't get the tags on in time, what can we do? So here's a variety of products that can be used for late season fly control. And so this will be in the documents you can get from the box link. If you don't need to write all these down, I'll provide all this later on. There are some new face fly control products. So one is a Trizap. So it has a permethrin and abamectin and a piperonyl product. These have a label to control face flies for 2 months. Again, David Boxler at our North Platte Extension Center did a lot of work and field trials on these and showed a 70% reduction from 9.96 flies per calf to 2.96. And you can see more of the details on his research on fly controls at beef.unl fly control. He's got a webinar that's really good and goes into a lot more detail. This was one of my maybe future research projects. If anybody wants to help me out, if you got some black cattle, you want to paint zebra stripes on them. Some researchers in Japan have showed that showed reducing biting flies attacks, but I haven't had anybody take me up on trying this out yet. But I guess if you have some white paint, maybe it's something to try. Okay, so besides flies, what are some other risk factors that we have? Again, dust and aerosolized particulate matter, UV radiation. This promotes the type of cells. It irritates the eye and promotes the cells that those bacteria can really stick to. Pasture types, white-faced cattle, genetics. And we mentioned nutrition and the body condition scores of those cows. And so there's some work that shows that copper, selenium deficiency may be associated with the risk of pink eye outbreak. Okay, so how do we treat pink eye? So we have two labeled drugs. These are tlathromycin and oxytetracycline. You can be able to consult with your veterinarian if there are other options that might be available. The one thing I want to stress is that timely treatment is really our best solution for pink eye outbreaks. We can reduce the healing time by at least a week and typically 2 for those ulcers if we get them treated rapidly. So if we get one of those drugs in there, we get the toxin and that bacteria eliminated, we're going to reduce healing times by up to 2 weeks and maybe more. And just remember those pictures I showed you that, an ulcer takes at least 2 months to heal if it's severe enough. So keep that in mind, that it's not a response failure. It's just that healing process. What we should avoid is we don't want to use dry cow intermammary tubes. We don't want to, there's other home remedies. I've heard all sorts of things from salts to WD-40 to oils. Please don't use those. Those are painful. They damage tissues. They're gonna cause more problems than they're worth. There's lots of welfare concerns. They're gonna damage tissue and delay healing. Puffers used to be commonly recommended, but those nitrofurazone drugs are illegal in food animals. You don't want FDA coming and knocking on your door, so please avoid those. Some other things that work great are patches and flaps. I'm not very good at subconjunctival flaps. Mine always loosen and tend to rub on, so I don't do them often, but I think if your veterinarian is good with them, they can work. The easiest thing is patches. So some new work by Gabby Meyer, she's the extension veterinarian at UC Davis, showed that just putting a patch on there decreases healing time by 4 days. So if they used a patch, those ulcers healed in 10 days versus 2 weeks in the controls. So patches, we now have a lot of strong evidence, scientific evidence with randomized control trials that these work great and reduce healing time, and they're easy to use. You don't have to buy the expensive patches. You can go to Goodwill or buy a bunch of old jeans, cut those up. Just some denim fabric with some tag cement works great. So we did on the prevention side, we did a vaccine trial in eastern Nebraska. This paper is freely available through the journal Vaccines to anyone. You can download it. But it was a 5-year randomized controlled trial. And we had to do 5 years because you can see we only got 3 years where we had enough pink eye that it was able to do any sort of the math on our vaccines. So 2 of those years we didn't have any. What we saw was we had a decreased incidence. We had 3 different formulations. So autogenous, that was one that we made with strains from our herd. So we isolated those and sent those to an autogenous vaccine manufacturer, and they made it for us. We ordered a commercial vaccine that was Moraxella bovis, Bactrin, and a sham, which was just the vaccine adjuvant. And we administered those twice and those calves were about 60 days of age, and then followed them throughout weaning. We found that the autogenous vaccine, we were able to generate significantly higher antibody levels, but those antibody levels didn't translate to protection. So you can see we had high levels of antibody in our autogenous. We had we had pretty good levels in the commercial compared to the sham, but those didn't translate into protection. You can see that our cumulative incidence, our lowest incidence was in the autogenous, where we had 24.5% Pinkeye. The commercial had 30.6 and the sham had 30.3. There was a trend downwards for the autogenous treatment, but when we ran the statistics, there wasn't a statistically significant difference. So we're still going to continue doing research and try to figure out why that might be. But in our field trials, we weren't able to sort of move the needle on the Pinkeye incidence by altering up our vaccine strategies. One issue may be timing. So we heard some talk about timing. So it's really important that we know when Pinkeye happens in our herd, so we can try to maximize the timing of the dose to maximize immunity when that's going to occur. We tend to see these IBK outbreaks later, so September, October, and most of our vaccines are given in May. So that may be something we could help, increase the immunity going into the highest Pinkeye risk. We know our pink immunity is going to be a week or two after that booster dose, and then it's going to start to wane. So if you kind of know when you see Pinkeye, it might be useful to start 6 to 8 weeks with your vaccine series when you normally see cases, and then follow the label for the administration. So the timing may be something that could be altered to increase the efficacy. Another thing we're working on, and maybe why our vaccines don't have the efficacy we want, is that these bacteria have really tremendous genomic diversity. So we've sequenced whole genomes of both bovis and Moraxella bovaculi. We found there's really tremendous diversity in these bacteria, and so if you're trying to chase what you got last year with a vaccine, or use a vaccine that may have historical strains, it can be really challenging. The interesting thing is that we found 2 distinct genotypes in both these types of bacteria that tend to associate with Pinkeye. So we have sort of one genotype that we find in normal calves and another one that we find during outbreaks. And you can see that the ones we find during outbreaks are much more genomically diverse. So they have several branches coming, all sorts of, if each one of these branches indicates how much different they are from each other, you can see there's a lot going on there. Where the ones we find in normal calves are not as diverse. So there's something making these move, and it may be things like vaccines or immune responses that are pushing them around to try and avoid that immune response. So just like kind of we hear with things like coronavirus, right? The vaccines need to keep up with what's actually circulating in the animals. Okay, so our take home, IBK or Pinkeye is caused by Moraxella bovis. Experimentally, we can give that to calves and reproduce disease. All the other pathogens, we have not sort of been able to do that yet, so it's complicated. Face flies are a major contributor to transmission. Our diagnostics show high rates of isolation of Moraxella and Mycoplasma in Pinkeye samples. These are both opportunistic pathogens that normal calves carry in their nose, and that amount of those that carry them varies by age. So there needs to be some sort of environmental or host factors that make those cause disease. The genetics of these are diverse. They're undergoing genomic pressures and changing all the time, and so it can be hard to keep ahead of that, the bacteria. And then the final take home is that prompt treatment leads to reduced healing time. So we can knock off 1 or 2 weeks of healing time if we get those treated quickly and appropriately. Remember that even if we do get them treated, they can take weeks to months to heal. So that's not a treatment failure. That's just the eye healing itself. And then patches can significantly reduce healing time in California.
I do have a question for you already.
All right.
This first question says, if the vaccine increased antibodies, could multiple or higher quantities contribute to actual Pinkeye prevention?
Yeah, so we don't think so. We're seeing this gap sort of no matter how much or when we give. There doesn't seem to be a relationship between the amount of antibodies and the rates of disease. So what we're focusing on is if we can get the antibodies more specific, so that make better antibodies, then more of them. So if we can find better antibodies and better targets, that's kind of where we're looking at in the future of our research. So that's a great question.